Hi, welcome to our lesson on cardiac muscle. This will be your first lecture on a lesson on muscle in general. So I'll be doing a little bit of introducing muscle and covering some of the general aspects of how muscle works, along with the things that are specific to the cardiac muscle. So first of all, there are three kinds of muscle: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle connects bones via tendons and causes movement in joints. It's important for you to recognize that it is different from cardiac muscle and smooth muscle in that it contracts as a result of the transmission of a signal through the neuromuscular junction. That is, an action potential starts in your brain, where your brain goes, contract that muscle. It is propagated down the nerve, and then at the nerve terminal, the signal is transported across a synapse, the neuromuscular junction, which then initiates contraction of skeletal muscle. So skeletal muscle is completely voluntary in that your brain says, hey, do it, and then you do it. It is striated, which means that if you look at it under a microscope, you can see bands under the microscope, and that would be as opposed to smooth muscle, which I'll talk about in a second. Cardiac muscle is, it forms the heart, of course, and it is involuntary in that your heart beats no matter what. You don't have to think about it consciously. You don't voluntarily contract your heart. It just does it sort of automatically. And it is also striated. Unlike skeletal muscle, there are a bunch of individual cardiac muscle cells that are lined up one after another, and these are electrically coupled through gap junctions. And whereas skeletal muscle cells tend to be very long and more or less run the length of the muscle, in some parts of the heart, for instance, the sinoatrial node, the cells throw action potential spontaneously. That is, they have pacemaker activity. And this would be unlike skeletal muscle in that skeletal muscle does not, normal skeletal muscle does not ever spontaneously generate action potentials. And as you know, this is of course how the heart rate is set and generated. It's, the cells in the SA node spontaneously throw an action potential, and then that action potential propagates from cell to cell and causes the heart to contract. Smooth muscle, constituent of many organs. Smooth muscle is kind of tough to describe in that it's so different everywhere in the body that you end up talking about very, very general characteristics. We'll worry about that when we do the smooth muscle lesson. It has the same basic contractile elements as cardiac muscle and skeletal muscle, but they're not very well organized, and that tells you that it's not striated muscle. The organization of the myofilaments, the organization of the muscle gives these two their striated appearance. Smooth muscle is unstriated in that it's not as well organized, and they can display spontaneous activity, again, unlike skeletal muscle. So a very common way to teach these tissues is to compare and contrast them. We'll be limited in our ability to do that in that we aren't really going to cover skeletal muscle in this block. There are certain aspects of it which I will be bringing up though, and you should be aware of these differences between the cell types and whatever it is I highlight as we go through the lessons. Okay. So these are cardiac muscle cells. This would be a typical one. It is a typical cardiac cell would be about 100 microns long by about 20 microns in width. And these cells are connected together with intercalated discs connecting them in between. And this is an intercalated disc that you see here. There are two types of connections in these intercalated discs. One is desmosomes. these are basically anchors which hold the cells together, and they're structural elements. That's pretty much all they are. And then we have gap junctions, which are pretty big channels that connect these two cells together. And small organic molecules and ions can flow from one cell to another bidirectionally through these. And you can propagate action potentials from cell to cell because these exist. And I've described this already in the introduction to the cardiovascular system lessons focusing on the heart. So I won't go into any great detail, but this is how these cells are electrically coupled to form a functional syncytium. Okay, this is the sarcomere. It is the basic functional unit of cardiac muscle. If you were to look at a cardiac muscle cell, you would have a lot of these sarcomeres lined up end to end along myofibrils, which are bundles of myofilaments that run from end to end through the cardiac muscle cell. So these myofibrils are, and of course the sarcomeres sort of making up these myofibrils, are made up of thick filaments, which basically is myosin, and that is sort of these darker lines that you see here, running through the middle of the cell. So you can see we're lighter here, we're lighter here. These darker lines are made up. That's where the myosin is. And they are bigger myofilaments than the thin myofilaments, which are made up of actin, tropomyosin, and troponin, and we'll go into more detail on these later on in the lesson. But you can see how these myofilaments are organized. They are lined up in parallel, and they sort of overlap. So the thin filaments overlap the thick filaments, and you can see the overlap there. So this is a little bit of a closer look at these sarcomeres and the myofilaments as they're lined up. So this dark band here that you see sort of in the middle of the sarcomere is the A band, okay? And the A band is basically where the myosin is, as I said before. Your myosin filaments in this sarcomere basically go from here to here, your myosin myofilaments. All right, and so that big dark band is basically the myosin. You can see the myosin here, kind of a heavy line here, and it runs the length of the A band, okay? We have the I band in the sarcomere, and this is just actin, okay, no myosin. So this is myosin-free actin. It's actin myofilaments where there is no overlap with myosin at all. That is your I band, okay? Your H band is actin-free myosin. So here's your A band with myosin, okay? And you'll notice that it gets a little bit lighter here and here and here. This is your H zone, okay? And so this is made up of myosin, but with no actin overlapping. So the actin overlaps with the myosin, okay? And it does that on the outer edges of the A band, but here in the middle, you run out of myosin, and all you have, or you run out of actin, there's no overlap, and all you have is myosin. That's where your H zone is, okay? And finally, you should know the Z lines, which sort of border the sarcomere here and here. And these are proteins which anchor the actin. So the actin connects to other proteins at the Z line, and those proteins at the Z line sort of hold the actin in place, okay? All right. And your M line. Your M line also has proteins which anchor the myosin in place. So it's this slightly darker band running right through the middle of the H zone. There are proteins there that are holding that myosin in place so that it doesn't just fly all over the sarcomere and move laterally or or transversely through the cell. Okay? All right. That is the end of my sort of introductory video on cardiac muscle, and we will move on to the next lesson as soon as you're ready. Thank you. Or the next video as soon as you're ready. Thank you.